So I would like to first thank the organizer to allow me to present this work. So as I say, I'm from the Genome Institute of Singapore, and this talk will be about the project I've been working on for the last 2 years, and it's actually almost finished. And it is how we use hybrid assemblies to actually explore the microbial diversity into Singaporean gut metagenomes. So I will try to summarize most of the most important result for this paper, but obviously there would be much more to actually say about it. So as I will say in the end of the presentation, there is a paper available, so feel free to look at it. There is more results inside it. So I will just make a very short introduction. So as we all know, the microbial diversity is vast and remain partially unexplored, and there is a lot of different reasons why there is this parts of the microbial diversity we're still missing, and this part is actually called dark matter by some people. And some of the reason is, first, we need a large number of samples. This is a larger number of samples, and this is like the obvious answer, but there is other reasons such as sampling bias. So if we look at, for example, gut microbiome, for example, it has been for a very long time limited to Western cohorts. There is so much different microbiome which can be explored in other parts of the world. And for not only gut microbiome, but for all of the different microbiomes analysis, it's interesting to look at other cohorts. Among the other reasons is more technical. It's like short-read assemblies, which have been used for most of the metagenomic studies before, leads to fragmented and error-prone assemblies. If you look on the right there, it's a very simple graph showing the depth of coverage required to link to the contiguity of an assemblies for a simple equal assembly using Illumina reads. And you can see that you need at least 30 to 30x coverage to provide a decent assembly for E. coli. So for an isolate, it's quite simple. But when you talk about metagenomic assemblies, having 40x coverage, especially for the low abundant species, is quite difficult. So this leads to fragmented and error-prone assembly, which makes the whole analysis difficult. And in my lab, we've been exploring new ways to resolve this missing metagenomics diversity and to improve the genome assemblies we can get. And it's not so recently, but 4 or 5 years ago, we published this hybrid metagenomics approach. It's software called OperaMS, and basically what it does is it's using the short reads to generate high quality assembly in a regular way. And after that, it's using long reads ONT to actually scaffold different contigs you have based on the short reads. There is more about this. It's not only doing that, but I will not give more details into this presentation. So the next step after publishing this in my lab, I don't know why it's doing that, sorry. The next step after was to how can we use these hybrid assemblies to actually generate high quality reference genome code for a metagenomic, for a population. So in our case, we've been looking for to the Singaporean population, where my lab is coming from. And Singapore is very interesting because it's a multi-ethnic population there. It's composed mainly of 3 different ethnicities: Chinese, Malay, and Indian. And so what we did there is we used 109 samples from healthy gut people, and we sequenced the gut metagenomes using both short reads and long reads, and from all of the different ethnicities. And the goal of these studies is double. The first one was to show the interest of using a hybrid assembly, and the second one is to actually show the specificity of the Singaporean gut metagenomes. So kind of a fishing expedition. So as I said, we did Illumina and nanopore sequencing for 109 samples allowing the hybrid assemblies. We also did high-C sequencing for 24 samples. I will just have one single slide. And so the first goal was to show that the interest of hybrid assemblies. So we did assembly for hybrids and also the assembly for only short reads for the 109 samples. a very classic binning and evaluation approach. And the first thing we can see here is that hybrid assembly first provide more max compared to short-read assembly. So you have a bit less than on average 40 max per sample using hybrids, while for short reads it's around 25 max per sample. But it's not only a number of sample, a max per sample which is improved, but also in terms of contiguity. So you can see like when you look at the N50 on average, it's much higher using hybrids compared to short reads. Okay, I'm sorry, I'm just gonna very quickly change this because I don't know why it's doing this. Okay, I'm very sorry. I don't know why it's doing this now. So we have a 25-fold improvement in terms of assembly contiguity looking only on N50 when we have hybrids compared to short reads. And if you look at what is hybrid-only max, it's actually, you can find them in all of the different genera. And interestingly enough, the abundance of this hybrid only is not only low abundance species, but it's also highly abundant species, for example, in the bifidobacterium. So this means that when doing assemblies using short reads, there is very highly abundant species, up to 25% abundance, which are missing an assembly using short reads only. After that, we classify the different mags with a very classical approach with the mi-max standards from medium to high quality. And what you can observe that is that using short reads, we have absolutely no high quality mags, while when using the hybrids, you have almost 20% of them, which is defined as high quality. And interestingly, when we look at this high quality and we compare that to what is available on public database, GTDB in this case, we can see that we can improve from medium to high a fair enough number of mags. And interestingly, it's not only uncultivated, but also isolates. And we also have an improvement of the contiguity of these mags, especially for the uncultivated one. Just a very, very short slide about the HiC data. So we did HiC sequencing, contact mapping for 24 samples using the Phase Genomic company pipeline. So it's really only binning in this case. So the HiC contact mapping helped to bin the hybrid contigs we have before. And what you can observe using this data is that the contiguity doesn't change. We have a slightly improvement of the number of mice going from 924 to 1043. But what is more important is that the fraction of high quality really increased using the HiC data. So the HiC data improves the overall max quality. So the improvement is not very significant when you look at contiguity, but if you're very looking for very high quality mags, it's actually interesting to do HiC. So this was for the comparison between short and hybrid assemblies. And the next slides are going to be what is unique about Singaporean population. So when we have done the verification of all of the mags, we found out there are 685 species. And when we compare them to the public database, we found them that 10% of them are novel, which is not such a bad number, not a small number, I mean, because gut metagens have been extensively studied in the past. So finding 10% of the species, almost 70 of them, sorry, it's quite high, and it showed clearly that there is some uniqueness into a Singaporean and potentially South Asian population. And when we look at the richness of the sample and making a rarefication study, we actually found out that we have potentially an estimation of 736 species for this community, and we're not so far from these numbers with our study, showing that if you sequence 100 samples with our technique, you're most likely going to have a very already nice representation of what is present into your sample. So using these high quality genomes, the second things we wanted to do is can we go a bit further about novelty? So we compare all of the genome to what is already provided with a very similar approach, but just with a more restrictive divergent to 1%. And we found out that most of the genomes we have are actually novel, 87% of them, including into very well-known genera such as bifidobacteriums and Faecalibacteriums. So it's very interesting because it shows that there is potentially a large amount of uniqueness in terms of genes or pathway, at least in genome content into these strains compared to what is available. So it's not only a novel strain, a novel species, it's other like novel strains and novelty in terms of functions. So obviously the next step was been to explore these potential novel functions. So Dr. Chia Minghao in our team have been looking at this, and he's been looking at biosynthetic gene clusters using two different ways, anti-smash and deep BGCs. And what he found out is there's a very high number of BGC which are not found in existing annotations, so it's quite novel, 88% of them. And if we group the BGC to gene cluster family, this number is actually even higher. It's 94% of the GCF which are considered as novel in all of the different categories. And we wanted to see like maybe some of these BGC have a very important functions, obviously. So we've been lucky and found a very nice case with the bacteriocins. So bacteriocins are toxins which inhibit bacterial strains growth. So it gives a competitive advantage over for some strains over or some species over some of the strains or species. And so we found out that one of these bacteriocins into a Bloschia actually is linked to a very strong association correlation to other species, especially Faecalibacterium. So you have this Bloschia species with the bacteriocin, and you can see the red link showing a very negative interaction with them. But if you have the same Bloschia but without the bacteriocin, you don't see this negative interaction. So we hypothesized, we didn't confirm, but we hypothesized that this bacteriocin may have a very highly impact to the microbial configuration in our community. So it's a very nice example of what we can find using this BGC approach. So I'm going to finish my talk with this. So it's been quite quick, but I hope that with these slides, I already give you some proof of the interest of doing hybrid assemblies in order to generate very high quality genomes and potentially a catalog for a community. So it's not only the identification of new species and new strains, which is important here, it's also that making this high quality allow you to improve the subsequent analysis, such as discovering of gene, BGC, or pathway. And we, in our mind, this approach is the first step of and the foundation of more which can be done. So once you have done this very high quality reference catalogs, this one can be used for a more extensive cross-sectional studies where you use only short-read assemblies, or for, in other case, for example, clinical studies to look at very specific pathway. And obviously, last but not least, I hope that I show with this work that there is a very interesting unknown diversity which can be found into a unexplored community, and in this case, Asia, but more specifically South Asia, and that we can really find unexplored microbial diversity in terms of species, but also in terms of functions. So I will just finish with some acknowledgement. So obviously, I would like to thank all of the people who've been working on this in my lab, especially Niranjan Nagarjan, which has been my PI, which is still my PI, by the way, and some collaborator, NUS, from NUS, so the National University of Singapore. Yiking Teo have been the one behind the initial cohort where the sample have been collected, and Henning Sidorf have been helping into the sampling as well. So the paper is already available in bioRxiv, and it has been recently accepted in Nature Communications. So hopefully we will have a very nice version of this study available into the journal very soon. Thank you very much.